All right, so hopefully you've watched the first one of these videos before we get back onto before we move on to the second one. But this lecture is going to really discuss in a lot more detail the actual regulation of the Lac Operon. So it's kind of the second part of 14.2, section 14.2 in the book. So just as a quick refresher on what we're talking about. We're talking about the Lac Operon, which has these different parts to it, right? It has these three structural genes, Lac Z, Lac Y, and Lac A, that are all instructions to make three proteins. They're all transcribed together because they're all required for the metabolism of lactose. Lac Z contains the instructions to make beta-galactosidase, which cleaves a lactose molecule in two. Lac Y encodes basically a membrane channel that allows lactose into the cell, and Lac A encodes this galactosidase transcellulase that we're just not going to worry about for the time being. The Lac Operon also contains three distinct DNA sequences that act as binding sites for regulatory proteins. The cap site, which is going to bind this CAMP binding protein that we'll talk about later. The Lac promoter, which binds RNA polymerase, and the operator site, which binds the Lac repressor. And please remember that the Lac repressor is a DNA binding protein that is made by this regulatory gene called LAC-I. And that gene is constitutively active. It just churns out Lac repressor all day long, and indeed all night long. So how is this important operon regulated? So what you're looking at now is a slightly different schematic of exactly the same thing, right? This time we've made the DNA control regions, the regulatory regions, bigger so we can draw the proteins as they bind to them. But you still have the same Lac operon with three structural genes and the three DNA binding regions as well. So please remember the purpose of this operon, right? It's going to make these proteins that are required to digest or metabolize lactose for this bacterial cell. When there's no lactose around, you don't want to be making these genes because there's nothing for them to do, and it's a big fat waste of energy to go around synthesizing proteins that you don't need. So this is an inducible system. The way that this particular inducible system works is that there is a repressor. We already know about this, right? It's made by this gene LAC-I. So this gene LAC-I is constitutively active, constantly making LAC-I mRNA, which is constantly being translated into this Lac repressor protein. This is made all the time. And what this protein does is it binds very specifically to the LAC-O DNA sequence. So it literally finds its spot. If my arm was a piece of DNA, right, and my hand is the Lac repressor, it finds the spot that it is designated to bind to simply by the DNA sequence that's present, and then it binds to it. And it'll sit there on the DNA. And what does it do? Well, the Lac promoter is just upstream of the operator. So it's away from the structural genes. And the LAC-P, the promoter's job, is to bind RNA polymerase. And it's shown in that state here. So we have RNA polymerase bound to the promoter. But notice that the RNA polymerase can't do what it wants to do, which is to move down this piece of DNA and transcribe these genes. And it can't do that because there's a whacking great repressor protein right in its way. And that's literally how it works. It can't move past that protein because it's in the way. It's actually called steric hindrance, but it doesn't really matter. What really matters is that basically the presence of the repressor blocks the movement of RNA polymerase and stops it transcribing the Lac operon. So in the absence of lactose, this is the situation, which is most of this cell's existence, right? So for most of the time, this operon is not transcribed. But if this DNA is present in a bacteria in your gut, and all of a sudden you eat a pizza, then you've just ingested a whole bunch of cheese. You've just sort of flooded your gut with lactose, in effect. And now this bacteria wants to use that lactose, wants to break it down and use it for energy. So it wants to then transcribe and translate LacZ, LacY, and LacA. So somehow it wants to remove the repressor from the DNA to allow RNA polymerase to move down this operon and transcribe this gene. That means it's got to basically bind to some kind of inducer molecule. And the inducer in the case of the Lac Operon is something called allolactose. Now, allolactose is basically levels of allolactose in the cell are going to increase when lactose is outside of the cell. So basically, this operon is activated by the presence of lactose, which is exactly what you want. And it's super direct. Basically, a byproduct of the lactose molecule itself binds to the repressor protein and then induces a three-dimensional conformational change, as shown in the bottom left of this panel, which prevents it from binding to the DNA. No repressors bound. RNA polymerase can move along the gene just fine, or the operon just fine. And in a nutshell, that's how the Lac Operon works. In the absence of lactose, a repressor is bound to the operator. In the presence of lactose, allolactose binds to the repressor, causes it to fall off the DNA, allows RNA polymerase to do its job and transcribe this operon. So as we've mentioned before, two of the gene products of the Lac Operon are lactose permease, made by LacY, beta-galactosidase, made by LacZ. Notice that when lactose gets into the cell, it's going to be split into two by beta-galactosidase to use as energy. But there's this side reaction that I talked about previously in the previous lecture. And look what that makes. It makes allolactose, which is what induces the Lac Operon expression. Now, one thing that might be confusing to you, in fact, let's talk about on the next slide, but one thing that might be confusing is that I said in the absence of lactose, right, you don't get, you get a repressor bound to the operon and you don't make the proteins. What that would mean, right, is that you wouldn't make lactose permease, which would mean there wouldn't be a way to get lactose into the cell. And if you can't get lactose into the cell, how are you going to make allolactose, which is going to induce this operon? And the answer to that is an important one that kind of helps you think about these cells in a more accurate way. And here's the answer. So in the absence of lactose, Lac repressor is bound to the operator. That is true. However, cells are extremely dynamic. And if you think about how biology works, you need a lot of flexibility in the way a cell operates, right? So if this Lac repressor bound to this operator in a very, very strong way and was very hard to get off, that kind of wouldn't make a lot of sense because it needs to be very responsive. So the way this actually works when there's no lactose around is that the vast majority of the time, there's a lot of Lac repressor in this cell, and there will be some of it bound to the operator, preventing expression of this operon. But it doesn't bind perfectly. So every once in a while, this repressor is going to fall off. And pretty quickly, it will be replaced by another repressor, because remember, repressor is being made all the time in this cell. For that brief moment when it falls off, that means some RNA polymerase can escape and make some of these proteins, or or transcribe some of these, transcribe the operon, basically. Pretty soon that's shut down when another repressor binds, but that sort of leakiness in the system allows a little bit of these genes to be expressed all the time, which means there's always a little bit of lactose permease made and a little bit of beta-galactosidase made, which allows the cell sort of a sensing system for the presence of lactose in its environment. And so that's how this works. And so let's look at what happens let's look at what happens when you drink a glass of milk to the bacteria in your gut. So here's a cell that's sitting there in your gut with no milk or no lactose in its environment. As I said, the Lac repressor is bound to the Lac operon in this case, but occasionally some RNA polymerase will escape to make small amounts of the proteins, for example, galactosidase, galactosidase transcellulase, lactose permease, and of course, beta-galactosidase. So because you've got these low amounts of these proteins, if you do drink that glass of milk, which is shown by these little gray dots, all of a sudden, this cell is bathed in lactose. Some of that lactose can get in the cell because there's some of these lactose permease proteins sitting around on the surface of this cell. So here's lactose, it's coming in through these little gates. Some of it will bind to the Lac repressor, causing it to fall off the operon, which then of course allows massive upregulation of this operon, causing the cell to become flooded with beta-galactosidase and also transacellulase. And there's going to be lots more permease expressed too, right? Because those three genes are all expressed at the same time. So here we have now tons of permease allowing tons of lactose in, allowing the cell to use that for its energy source. So great. But like I said, unless you're mainlining dairy 24/7, lactose is not always going to be around. And pretty soon, your gut is going to be free of lactose, and there's going to be no more for the cell to use. So what happens then? And how is the operon shut down to get it back to its normal repressed state? Well, the lactose is going to go down outside the cell as it decreases in the environment, and it's going to go down inside the cell as the cell uses it for energy. So eventually lactose becomes depleted. That means that levels of the inducer, allolactose, are also going to go down, which means there's less inducer to bind to the repressor, which means all of a sudden there's a greater chance that the Lac repressor is going to be on the Lac operon. We have all of these spare enzymes lying around, of course, and these spare channels on the surface, but there's a lot of flexibility in cells. There's a half-life to proteins. They eventually go away. They become degraded over time. And unless you make more of them, levels in the cell are going to fall. And that's exactly what happens here. So most of the proteins involved in the use of lactose are degraded. They fall apart. They're recycled, which gets us back to the initial state where we have low levels of the enzymes in the cell and low levels of lactose permease outside the cell. Now, of course, that situation is not going to carry on until there's none of these proteins left, because as I said, there's a very small amount of each of these proteins made, even in the absence of lactose, because the Lac repressor will sometimes fall off the DNA and allow a little bit of these proteins to be made. And so that's essentially the cycle. It begins with you drinking a glass of milk, flooding the environment of this cell with lactose. A little bit of lactose gets in through the permease, binds to the repressor falls off the operon, you get a massive upregulation of permease, and the other structural genes, allowing the cell to use that lactose as an energy source. And then eventually it goes away and the proteins degrade and you get back to where you were in the first place. And that's this inducible system, right? In that absence of lactose, the system is repressed. It becomes induced in the presence of lactose. And then when lactose goes away, it becomes repressed again. And so it's pretty smart, right? It's pretty efficient. You're using the thing that you're going to metabolize as the inducer of the enzymes that are going to metabolize that thing. So it's very efficient. And that's the Lac Operon and how it works. There's another aspect to it, though, which is another important, well, actually, so one more thing I'll say. So this idea that you have a repressor that binds to the DNA that is then somehow repressing transcription, is then released from the DNA, that kind of thing, very common in transcriptional regulation in both eukaryotes and both prokaryotes too. But it is also often true that for any given gene, you want a sort of sophisticated way to regulate it. And there's a good example in the Lac Operon of that occurring. And it all revolves around that third DNA binding site that we didn't really talk about yet, but we will now. And that basically solves this following problem. So if I'm in fact, any cell, not just a bacterial cell, but almost every cell, glucose is your primary source of energy. It is directly entered into cellular respiration, as you guys know from your cell bio studies of that process. So glucose is a better energy source than basically anything else, including lactose. So if I was drinking this cup of tea and it had milk in it, but it also had glucose in it, then I'm going to flood the cell's environment with two sugars that it could use, lactose and glucose. Now, given the choice, the cell would prefer to use glucose over lactose for energy. This is an example of something called dioxic growth, which just means the cell can choose to use different nutrients to grow, basically. So because of that, when you have a lot of glucose and a lot of lactose present, glucose, there is a way that the cell basically does not express any of the genes in the Lac Operon when glucose is present, no matter if lactose is present or not. And this is an example of catabolite repression. So glucose is a catabolite. It's something that the cell can catabolize to live. So is lactose. But in this case, the catabolite glucose is going to repress the genes that are required for the metabolism of lactose. How does this work? To understand how this works, and really kind of the most difficult part to get your brain around, is that there is a small molecule in really every cell in biology that's called CAMP, cyclic AMP. And in the case of bacterial cells, this is just a small molecule that floats around on the inside of the cell, and it kind of acts as a monitor for glucose levels. And then if glucose levels are high, it will help shut down transcription of the Lac Operon, because you want to use glucose rather than lactose as your energy source. Now, when glucose is present, and this is the bit that you have to remember, when glucose is present, CAMP levels in the cell are low. When glucose is not present, CAMP levels in the cells go up and are high. The CAMP molecule combined to a protein called CAP to form something called CAMP cap, which is just them bound together. Now, CAMP cap acts as a great accelerator of expression of the Lac Operon. And that's kind of the opposite to Lac-I, right? So Lac-I is the repressor of the Lac Operon and kind of acts like a brake. So CAMP cap basically stimulates expression of the Lac Operon, whereas the Lac repressor, Lac I, inhibits. It acts like a brake. How does that all shake down when this is occurring? So remember this one fact, or I guess these two facts. One fact is that when glucose is present, CAMP levels are low. They're driven down in response to glucose. And when it's not present, they go up. So look at the top part of this slide for a second, and you'll see what happens. So the situation is shown on the right. Lactose is present, no glucose is present. So what we've learned so far is that when lactose is present, there's going to be no Lac repressor bound, right? We've talked about that in detail. Now, I just told you that when glucose levels are low, CAMP levels are high. And what that means is there'll be a lot of this CAMP cap accelerator around in the cell because CAMP levels are high. So when that's true, here is the cap site, which is this purple binding site for the cap CAMP protein molecule. And remember, this acts like an accelerator. So if this is bound to the cap site, what it basically does is recruit more RNA polymerase to the promoter and cause the rate of transcription to go up. Now, if there's lactose present, there's no repressor in the way. So this leads to a very high amount of transcription of Lac Operon, which is what you want, right? Because there's loads of lactose around, but no glucose. So you want to use the lactose as your food source. Another situation with the cell could find itself is no lactose or glucose. Now, in this case, when there's no lactose around, the Lac repressor is going to bind to the operator and prevent RNA polymerase from transcribing this gene. There's also no glucose around though. So cap CAMP levels are high and they're bound to this cap site, but when the brake and the accelerator are on, just like when you drive a car, it doesn't move. And very low transcription occurs in this case too. What about the other possibilities that the cell can find itself in when it comes to lactose and glucose in its environment? Well, if both lactose and glucose are present, there's going to be low levels of CAMP because glucose levels are high. That actually prevents this transcriptional activator binding to the cap site. That means that the accelerator is off. Now, in this case, there is lactose present, which would lead to the repressor falling off the operator, but which would normally lead to transcription of this gene. But because there's no accelerator and no brake, transcription rate is low. Excuse me, going to go and shut up my dog. Okay, sorry about that. Dog dispatched. That's not in the way, that sounded bad. Just let him outside. Yeah, in the absence of, so when there's lactose and glucose around, the cell wants to preferentially use the glucose. And so in that case, because the CAMP levels in the cell are low, the inducer here is inactive. And even though there's no repressor, because lactose is present, transcription rate is low. Finally, when there's glucose present and no lactose present, then obviously the repressor is bound because there's no lactose present, but the cap CAMP is also not bound because CAMP levels are very low in the presence of glucose. And so the transcription rate is very low in this case. And so hopefully you can see that by using these two systems, one is an inducer, in other words, an accelerator, the CAMP cap, and the other one is a brake, the allolactose repressor system. You can preferentially use one sugar over the other, even when both of those sugars are present in the medium. So that's basically all I have to say about the regulation of this important operon. A lot of the general ideas here are used in transcriptional regulation writ large in prokaryotes and eukaryotes. Essentially, transcriptional regulation basically boils down to proteins binding to regulatory sites on DNA and either upregulating or downregulating transcription. There's lots of different ways that they can do that, but essentially the mechanism is basically the same. They just bind to parts of DNA and either turn genes on or turn them off. So I want to talk about merozygotes for a second, because this is going to be important to understand how you got to answer some of the questions on the assignment this week. So what you're looking at here, I want you to begin by looking at what is called a mutant strain of E. coli. So these types of experiments were done to try and figure out what types of molecules a repressor was, LAC-Z, LAC-Y. And the way they did these experiments was to take mutant strains of E. coli. So this mutant strain growing in this flask over here, if you look at it, this big circle is meant to represent the entire E. coli genome. And there are letters written on the DNA which correspond to certain things you already know about. And the key is on the left here. So I is the LAC repressor. So this is a gene that's going to make LAC repressor. It's not part of the operon, but it's important for operon regulation. P here is the promoter. O is the operator. So these guys are DNA regulatory sequences. And then Z, Y, and A are all of the three structural genes, LAC-Z, LAC-Y, and LAC-A. If there's a little plus by them, it means they're normal, wild type, operational, either proteins or binding sequences, and a minus means there is a mutant that stops it working. So one of the things that they did back in the day was they took these mutants. So this first one, right, is a mutant in LAC-I. So this has a repressor gene that doesn't work. So in this case, there will be no repressor in that cell at all, which would mean that effectively the Lac Operon would be on all the time, basically. There'd be no repressor bound, right? So ignoring the glucose regulation for a second, there's no repressor bound. It's on, at some level all the time. So that's what's going on in this mutant. There's no Lac repressor made. The Lac Operon is always in the on state. And then what experimenters did was they took a second piece of DNA that was a smaller piece of DNA, as shown by this little circle here. And they called this F prime, which just meant a plasmid. You may have talked about this in the lab portion of this class. I'm not sure because of our current situation. But basically what they did was they took a smaller piece of DNA, and by effectively combining these two strains together, they made something that they called a merozygote. Now, what that is just a partial diploid. So normally these are haploid cells. What you've done here is you've introduced a few genes, of which there are now 2 copies. And they know the identity of which genes were mutated on the F prime plasmid. And so they'd be able to say, hey, what gene can rescue what other gene? And in this case, what they did was they introduced a second plasmid or a second piece of DNA, this F prime plasmid, that is wild type across the board. And what they would find is that if you introduced a wild type repressor, this would rescue the mutant repressor over here. Because now all of a sudden the cell can make repressor, and repressor made from this piece of DNA is just a protein that's going to diffuse in the cell. It's got to bind to this operator and also this one, because it doesn't care. It just binds to DNA. And so you could rescue repression by bringing in a wild type repressor plasmid. And using this logic, you'll see when you do the assignment, they could try and tease apart essentially which of these components worked on the piece of DNA where the DNA sequence is, or on a different piece of DNA, whether it could move to a different piece of DNA. And what that means in the case of the Lac Operon basically is that, you know, these operator and promoter sequences, they can only influence what is right next to them. If I introduce a second piece of DNA, this promoter cannot rescue a mutated promoter on a different piece of DNA. Whereas something like a repressor that can bind to any piece of DNA can swap between pieces of DNA and rescue different phenotypes. Anyway, that's the idea behind a bunch of the experiments you're going to look at in your assignment. All right, so as always, if you have any questions, email me, post them on the forum on Moodle, and yeah, good luck and see you on the next one. Thank you for watching.